I'm going to ask next presenter, Jeffrey Now, Aviceda Therapeutics, to come join us.
Good afternoon, and thanks for the invite to the great meeting. What I'm going to talk about today is a new mechanism of action for addressing chronic immune disease, which is glycoimmune checkpoint therapy. When we think about geographic atrophy, we see this continuum of a chronic inflammatory process which exists in other diseases throughout the body, but we see environmental and genetic factors that ultimately lead to accumulation of toxic molecules. Then we have an invasion of the subretinal space with various immune cells. And so we need to really address this inflammatory component of the disease in order to be able to treat not only geographic atrophy, but to stop patients from progressing to neovascular AMD. When we think about what is the treatment approach that we want to bring to the table, we're really looking to inhibit pathogenic macrophages and microglia in the retina. We want to restore that physiological intraretinal and subretinal immune regulation. As we all sit here today, we have a homeostasis in our retina where we don't have chronic inflammation happening, and that's kept in check by these cells as well as our normal cells that I'll talk about in a moment. So we want to inhibit production of pro-inflammatory cytokines. We want to inhibit the pathologic phagocytosis that happens with macrophages, but also slowing the progression of retinal atrophy and then ultimately preventing the choroidal neovascular membrane. Going back to our cell biology that we were taught in high school, I want to kind of revisit this concept of the glycocalyx. All of our mammalian cells have glycoproteins on the outside of their cell membranes. These are important in maintaining that homeostasis. Sialic acid residues are the outermost layer of the glycocalyx, and these sialic acid residues bind to cyclic receptors that are on all of our immune cells. These cyclic receptors have multiple extracellular and intracellular domains. The extracellular domains interact with the sialic acid residues. The intracellular domains are inhibitory, such that we can actually bind these receptors and shut down the immune response. This is something that happens not only throughout the body on a normal basis, but also is a similar concept that's being leveraged to treat oncology indications. And when we think about how the immune system recognizes self versus non-self, it's done so by these cyclic receptors recognizing the sialic acid residues on normal, healthy cells. When we don't have the right glycan presentation, when we're missing sialic residues, when we have hyposialization, we get inflammation. And so it's really important that we're able to present those sialic acid residues to the immune cells. When we look at the receptors that are expressed on the various macrophages, microglia, and monocytes, we see there's some similarity, but also some differences. And so what we've developed here is that a glycan-coated nanoparticle, which contains really multiple different components, a bioerodible PLGA polymer core. On the outside of that is decorated with high-affinity sialic acid ligands. These bind to cyclic 7, 9, and 11, and it's important that they're oriented and have the right density. So we get this super self concept where it binds to the cyclic receptors, is able to shut down inflammation. When we look at the preclinical data here, we see we can not only reduce pro-inflammatory cytokines, we can reduce phagocytosis in both macrophages and microglia, but we can also stop monocyte differentiation from monocytes that are circulating in the circulatory system to activated macrophages. We've studied this product in a phase 2A single dose study with ascending clinical doses. You'll see here the baseline data, and you'll see these are large lesions with poor vision. And what we see is if we compare the lowest dose to the two highest doses, about 71% and 50% reduction in progression of geographic atrophy lesion size by autofluorescence. We can see about a 5-letter gain in vision in those top two dose groups versus the lowest dose group. And we can see that about 77% reduction in photoreceptor loss by ellipsoid zone assessment on OCT. From a safety perspective, we don't see much from a safety perspective here. We see one patient that had inflammation. This was resolved without treatment, and at the next scheduled visit, visual acuity was improved. So We see a product that is safe and efficacious, but again, single dose delivery in that study. We currently are studying this product in a multi-dose clinical trial, which is really one of the first head-to-head clinical studies against an active comparator, which is Eyecelerator. And again, here's our primary and secondary endpoints. We'll have readout this quarter of this study. Thank you.